So the outbreeding devices, right? So the outbreeding devices. So majority of flowering plants, they are hermaphrodite, right? And okay, so majority of plants, these are hermaphrodite. Okay, and uh, hermaphrodite flowers. Okay. Okay, so continued cell pollination result in inbreeding depression. So flowering plants have many devices to discourage self pollination and to encourage cross pollination. Okay, in some species like pollen stigma receptivity are not synchronized, so either pollen is released before the stigma becomes receptive, and like anther and stigma are placed at different position, and pollen and stigma receptivity are not synchronized. Now they prevent autogamy. Now the third device is prevent inbreeding is cell incompatibility. This is genetic mechanism to prevent cell pollen that is from the same flower or the other flowers of the same plant. Okay, from the other flowers of the same plant from fertilizing the ools by inhibiting pollen germination or pollen tube in the pistil. Now another device is prevent self pollination is a production of unisexual flowers. Like a flower can either be like male and female, so this is it. Okay, if both male and female flowers are present on the same plant as castor and maize, so these are monoecious plants. It prevents both autogamy but not heterogamy. Okay, now several species such as papaya, male and female flowers are present on different plants. So male and female flowers are present on different plants, that is each plant is either male or female, that is dioecious condition. This condition prevents both autogamy and heterogamy. Example is papaya, markensia. Okay, now pollen pistil interaction. So pollination does not guarantee the transfer of the right type of pollen, that is compatible pollen of the same species as the stigma. Okay, sometimes pollen of the wrong type also deposit, like now it is like same self incompatibility, right? Or compatibility, it's mean right type of pollen deposition, and wrong type, that is incompatibility, right? Now there is like okay, if the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. Okay, the ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. Now you so okay, so this is it, like uh, the growth, then pollen tube after reaching the ovary enters the ovule through the micropyle and enters one of the synergids through the filiform apparatus. Okay, now filiform apparatus present at the micropylar part of the synergy guides the entry of a pollen tube. Now all these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. Okay, now pollen pistil interaction, it is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen, right? So this is it, like now artificial hybridization, to get the superior varieties. So for this there is like uh, emasculation, then bagging, then dusting, and then rebagging. So emasculation, it is the removal of the anther from the female part. If the female parent bears bisexual flowers, it has both androecium and gynoecium structures. Okay, so removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is necessary. So this is emasculation. Okay, then bagging. so that the to prevent contamination of its stigma with unwanted pollen. Then once it receives the maturity, then dusting. Okay, the male parent are dusted on the stigma, and then the flowers are rebagged. Okay, now if the female flowers, they are unisexual flowers, if it has like only female part, so we don't have to worry about it. Okay, now there is no need for emasculation. Then flowers are bagged, then dusting, and then rebagged. So this is how artificial hybridization done to get the desirable varieties. Now double fertilization, right? So what about the double fertilization? It has syngamy and triple fusion. What is syngamy? It is like formation of embryo from the zygote, and a syngamy, it is the fusion of one male gamete with the one egg cell. And what is triple fusion? It is like two polar nuclei in the central cell, that is secondary nuclei gets fused with the another male gamete, that is triploid. So it formation of triploid primary endosperm. Okay, PEN, primary endosperm nucleus, that is 3N in ploidy. Okay, so this is like now also it develops into like primary endosperm cell, the central cell after the triple fusion becomes the primary endosperm cells and develops into endosperm, while the zygote develops into an embryo. So all I tell you now, there are some degenerating antipodal cells, degenerating synergies, zygote, primary endosperm cells, primary endosperm nucleus, that is 3N. So zygote, then globular embryo, then heart-shaped embryo, then a mature embryo. Now post-fertilization structure and events. Now post-fertilization, it means after the fertilization, like even following double fertilization, events of endosperm and embryo development, maturation of, so events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit, these are collectively post fertilization events. So endosperm and embryo also post fertilization events. Now endosperm, so endosperm development precedes embryo development, right? Why? Because the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue, and this tissue is like nutritive, like now they have the reserve food materials and used for the nutrition of the developing embryo. Now most common type of endosperm development is free nuclear endosperm, like the tender of the coconut has the free nuclear endosperm, and the white kernel, it is a cellular type of endosperm. Okay, now this is the coconut. Okay, now but the most common is the free nuclear endosperm. It is strictly not followed by the cell wall division like it. Okay, it is just like the division of the nuclear. The P and primary endosperm nucleus undergo successive nuclear divisions to give rise to free nuclei. Okay, and when the cell wall formation appears, then they become cellular. Now this is it. Now endosperm may be